Hello, everyone. My name is Bernd Meibohm from the University of Tennessee Health Science Center. I will today talk about the pharmacokinetics and pharmacodynamics of therapeutic proteins. Section 1 of the talk will introduce you to therapeutic proteins. The goals of this section are to appreciate the contribution of therapeutic proteins to the collection of approved medications, to understand key structural features of therapeutic proteins in contrast to small molecule drugs, and to recognize the role of clinical pharmacology concepts in the clinical use of therapeutic proteins. This slide shows the FDA drug approvals from 1993 to 2022 and differentiates between new molecular entities, or NMEs, shown in blue as traditional small molecule drugs, and BLAs, or biologic license applications, shown in gray. Biologic license applications include therapeutic proteins, but are also, for example, blood or blood products, as well as vaccines. As you can see, over the last couple of years, the number of biologic license applications from the overall number of approved drugs has relatively been constant as approximately 1/4. So over these last years especially, therapeutic proteins have become a major part of our collection of drugs that are clinically used. To remind everyone about proteins and protein structures, each protein has a unique structure, as everyone probably remembers. You usually have the primary structure based on the amino acid sequence that's shown here on the left side, where each letter stands for a specific amino acid. In addition to this primary sequence, however, you also have a secondary, tertiary, and quaternary structure. And this is basically the three-dimensional arrangement of this linear amino acid chain. So a perfectly linear amino acid polymer is neither functionally nor energetically favorable. In the aqueous environment in the body, you basically get folding based on these the interaction between hydrophilic and hydrophobic moieties in the amino acid sequence, and by that you get a defined three-dimensional structure. And this three-dimensional structure is stabilized by hydrogen bonding or van der Waals interactions, sometimes disulfide bridges, so relatively weak atomic interactions that produce a defined three-dimensional structure that is necessary for the stability as well as the function of the therapeutic protein. Since this stabilization is produced by relatively weak interactions, it also means that therapeutic proteins are usually relatively susceptible to disturbances from either mechanical stress as well as temperature stress. And by that, oftentimes, for example, require refrigeration for their storage. What I also would like to remind everyone about is that therapeutic proteins comprise a large variety of molecular structures. So you have structural diversities. As an example, a small therapeutically used protein is insulin, consisting of 51 amino acids and a molecular weight of approximately 5.8 kilodalton. Albumin itself probably not used as a therapeutic protein, but oftentimes a major part of fusion proteins. Albumin has a molecular weight of 66.5 kilodalton and consists of 583 amino acids. And compared to that, a immune globulin G molecule, like a monoclonal antibody, which has a molecular weight of approximately 150 kilodalton and consists of around 1,300 amino acids. As you can imagine, the different structure, the different molecular weight, and the different size of these molecules has also an impact on how they interact with structures in the body, how they are handled by the body, and how their PK and PD are affected. When we compare traditional small molecule drugs with proteins, then small molecule drugs are defined by chemical structure and purity. We know exactly what kind of chemical structure they have and in what purity they are available to, for example, be used as an API in producing a therapeutic therapeutic medication. They are chemically synthesized. Examples are just give me a fentanyl, ibuprofen, and they are identical from batch to batch. So since they are batch-wise produced and synthesized, these batches, if we compare them, are completely identical with regard to structure and purity. In contrast to that, therapeutic proteins are produced in living organisms in either mammalian or bacterial cells. They are defined by the production process rather than their structure, chemical structure and purity. So that then means that the production process needs to be extremely carefully controlled. It needs to be a controlled environment for these microorganisms, as they are usually defined in the chemistry, manufacturing, and control requirements, the CMC requirements for a therapeutic proteins. And then these therapeutic proteins that are produced are usually characterized with a variety of different so-called critical quality attributes, or CQA, that can be between 80 and 130 to 140 different attributes that are relevant for a large therapeutic protein like a monoclonal antibody. Each product is, through this defined production process, unique. Examples for therapeutic proteins, for example, adalimumab as a monoclonal antibody, or etanercept as a fusion protein. Since these proteins are also produced in batches in living organisms, these batches are highly similar, but they are not identical, simply because of small variations that may occur within this carefully controlled environment in which the genetically modified living organisms are grown, are housed, and ultimately produce the therapeutic protein of interest. So batches are highly similar, but not identical. When we look at major classes of therapeutic proteins that are clinically used, then the biggest one are probably monoclonal antibodies and antibody constructs. So this includes native IgG molecules like bevacizumab, used in cancer indications, infliximab for treatment of rheumatoid arthritis and other chronic inflammatory conditions. Then antibody drug conjugates, where a IgG molecule is conjugated with a small molecule drug that ultimately carries its own activity. So the antibody may either be used as a delivery device only, or may also contribute to pharmacologic activity. One example is brentuximab vedotin, used in lymphoma. So this molecule is obviously then produced not only by a production process that is in a living organism, but is then subsequently chemically modified through the conjugation process with a small molecule drug. And then we have more recently many antibody fragments and antibody constructs that use parts of an IgG molecule and combine it in a variety of different forms and fashions. For example, ranibizumab is used as an antibody fragment in age-related macular degeneration, or blinatumomab as bispecific antibody derivative used in acute lymphoblastic leukemia. The second big group are hormones and growth factors, which are usually identical or closely similar to endogenous molecules and supplement or replace a lack of these hormones or growth factors. An example, of course, insulin for type 1 diabetes, filgrastim as a growth factor for white blood cells to treat neutropenia, neutrophil growth factor, and then beta interferon for the treatment of multiple sclerosis. The third group are enzyme replacement therapies, where large molecules that have enzyme activity and that are either dysfunctional or completely missing in specific individuals that are affected by genetic disorders are replaced. An example for that is agalsidase beta in Fabry disease. Now, luckily, the central paradigm of clinical pharmacology that we know from small molecule drugs is equally applicable to therapeutic proteins. That means when we give a dose or dosing regimen to a patient that results in concentration time courses in different organs and tissues, that these concentration time courses are ultimately the driver for the clinically observed efficacy, as well as potentially adverse events or toxicity. And the relationship between the dose and the concentration is still characterized by pharmacokinetics, and the concentration effect relationship to the desired as well as undesired effects is characterized by pharmacodynamics. So we will in the following focus on the pharmacokinetics and pharmacodynamics of PK/PD of these therapeutic proteins. So in summary for this section, biologic license applications constitute approximately 1/4 of the overall FDA approvals for new medications in the last 10 years. Therapeutic proteins comprise macromolecules of wide structural variety with different molecular weights and physicochemical properties. Therapeutic proteins are produced in living organisms or cells and are defined by the production process rather than their chemical structure. And the same central paradigm of clinical pharmacology holds true for therapeutic proteins as for small molecule drugs. Here is a self-assessment question regarding the first section.